Okay, hi guys. This is Mitosis Part 2. So, we're back in chapter 12, and we're going to be going through the actual parts of the cell cycle and the phases of mitosis in this video. So mitosis is all about getting the right stuff. The reason mitosis is a complex process is because we have to go from being one cell where everything's organized and DNA's in the nucleus, etc., to being two cells that are identical to the original cell. So all the processes that we're going to talk about in relationship to mitosis is about organizing the stuff. All right, so that both cells have what they need to have. So if you remember the phase of mitosis, prophase, metaphase, blah-diddy, blah-diddy, then you remember that those processes are all about organizing the genetic material, right? So we're talking about what is passed on to daughter cells. And daughter cells is what we're calling the cells resulting from cell division. Right, so it's not daughter in the sense of not a son. It has nothing to do with being feminine. It's that we're calling the result of mitosis to be daughter cells. So you start out with an original cell and you end up with two daughter cells. And those daughter cells need to have the exact copy of genetic material, the DNA. It needs to be exact. So we're talking about, say, taking one skin cell and ending up with two skin cells. It's super important that it be the exact same material. All right, so this is about mitosis, of course, not meiosis. We'll talk about meiosis a little bit later in the year. So we're passing on the DNA. We're also going to be passing on organelles, cytoplasm, the cell membrane, and enzymes. And we're going to complete the process with cytokinesis. That's the division of the cytoplasm. And this picture that we're looking at is the chromosomes, this orange piece here, in a kangaroo rat epithelial cell. And I think somebody had to do the epithelial cell in their project. We have epithelial cell in a lot of parts of our body, but skin is an example of where there's some epithelial cells. So the point of this is to show you that you can clearly see the fibers of the cytoskeleton here as well. It's a beautiful picture, actually. Okay, so let's go over a little overview of mitosis, and then we'll go into detail. Okay. I'm gonna let them all be here, and then we'll talk about it. So this is to go along with, if you're following along in your book, page 222 and 223, and that's where they kind of go through it. So here we're looking at interphase. Okay, this is not a time of cell division, but it's just before mitosis is going to begin. So the chromosomes are already duplicated, but they haven't condensed yet. And you can see here, just for a point of reference, these are the centrosomes with the centriole pairs inside. And then we're moving on to prophase, where we have the mitotic spindles beginning to form, and the chromosomes have condensed, and the chromosomes are now consisting of the two sister chromatids. We'll go over that in a second. And then we have the Metaphase. Remember, this is a fluid process. It's not like these steps just happen. Okay, this is a whole fluid process. It's in motion, and I think there's an animation here at the end. So, oops, this has the names attached to it there. Prophase, Metaphase, where we're starting to be between the time when these have condensed and then the nuclear envelope's going to break down. Um, and then we're moving towards these guys. Remember, we're just, all these phases are about organizing the genetic material so that we end up with two daughter cells with the identical DNA. So we're moving on to metaphase, where the chromosomes are going to line up across the equator of the cell, and then anaphase, where they pull apart, and then telophase, where we're starting to get the nuclear membrane to form again. Let me push the button here. The nuclei are beginning to form. The envelope is forming here at the end. The chromosomes are starting to become less condensed, and we're going to move on to cytokinesis. The full division of the cytoplasm will happen in late telophase. It can also be pronounced telophase. You've probably heard both. Okay, and then there's the cytokinesis. All right, so this is how some people remember it. The interphase, prophase, metaphase, anaphase, telophase as IPMAT. I've had students make up their own system of remembering this. Paul makes a taco is one. So if you want to come up with something to help yourself remember it, if they're not already in your memory, please share it with us if you come up with something good. Right. So let's spend some time talking about interphase. And of course, for any cell, it's going to spend most of its time in interphase. All right. This is a cell doing its everyday job. Okay, whatever that cell does, it's going to be doing that for most of its time in interphase. And interphase is the whole time from it being a brand new cell till it divides again. Okay? So there are cells doing their jobs. They're making RNA, they're making proteins, they're making enzymes, they're making whatever they're making. All right, so they're working here. The cell is doing its regular job. All right, it will prepare for duplication if triggered. So it's going to have to be triggered to go on to divide, and we'll talk about that a little bit more. All right, so it's going to get the message to divide and multiply. So a cell has a life cycle, the cell cycle. So the cell is formed from a mitotic division. It's a brand new baby cell. Oh, it's so cute. It's a baby cell. Okay. It's going to grow and mature to divide again. All right, and then it's going to go through the stages of the cell cycle, G1, S, G2, and M. So let's talk about what those mean. Okay, people often mistakenly think that the G stands for growth, but it doesn't. It stands for gap. So this is the gap 1. This is the GO, the resting phase. We'll talk about that a little bit more. It's an important part. S is the synthesis phase. The synthesis phase is when the DNA is synthesized. So we go from having the original DNA to making a copy. We get into the details of that in another chapter and when we go over genetics. And then gap 2, and then the M for mitosis. So the cells that do this process, grow, mature, and divide again, are not every cell in the body does this. And remember that body cells are called somatic cells. But the particular somatic cells that are going to do this, okay, are the epithelial cells, blood cells, and stem cells. So not every cell in the body does mitosis, okay, but epithelial cells, blood cells, and stem cells do. All right, the other option is to grow and mature and never divide again. Okay, I'm sorry, I paused there, so I'm not sure exactly where I left off. So if I'm repeating myself, I apologize. Uh, so we're talking about somatic cells that mature and divide again, such as epithelial cells, blood cells, and stem cells. Red blood cells are kind of a weird exception that we'll get into when we talk about blood, because red blood cells don't have a nucleus, but your skin, the stomach lining, your hair, your fingernails, those cells are going to have the most rapid rate of mitosis. They're definitely going to be reproducing all the time. Some cells grow mature and then never divide again. So this is about brain and nerve cells and muscle cells. So these are really distinct cases that we're going to talk about when we go into anatomy, but this is why when you have a brain injury like a stroke, there are repercussions. The brain can be somewhat plastic, meaning it can recover in certain ways by compensating, but the tissue itself cannot be repaired. Whereas, as opposed to your skin cells, where when you have a cut and you've destroyed a whole bunch of skin cells, you're going to be able to repair that. And if it's not serious, you're not even going to see a scar or anything. The cells will go through mitosis and everything will be looking like brand new again. Whereas your brain, when it suffers an injury, that's not going to be the case. Even though you may have some compensation, the cells themselves are not going to repair. The unique case is liver, which after you've matured, your liver cells are not going to go through mitosis again unless there's an injury, and then your liver cells can go through mitosis again. So liver's a little bit unique that way. If you get too much of your liver cells destroyed, as in like the case of a chronic alcoholic or something, the rate of mitosis isn't fast enough to compensate for the amount of damage often, and that's how the liver can become too diseased to recover. So that just gives you some context, because we probably, in ninth grade tell you that all your body cells go through mitosis, but that's not really the case, okay? So the stomach cells, skin, hair, and fingernails are going to go through the most mitosis. Brain and nerve cells are going to go through it just until until growth has finished, and then that's that. And the same with liver cells unless there's an injury. We're going to talk a little bit more about the cell cycle after we go through mitosis too, because there's a pretty complex section you probably saw in the chapter about control of the cell cycle. So interphase is divided into the three basic phases that I told you. G1 for the first gap, which is growth. It's definitely called the gap phase, but you can remember that we're talking about growth. This is as we said, the cell doing its everyday job, and it can go into the GO phase, which we'll talk about in a minute. And so the cell is growing, okay, and it can receive a signal to divide, and that will take the cell into the next phase, which is the S phase, when DNA synthesis is occurring, and that's when we're going to get a copy of chromosomes, and then we move right into G2, the second gap phase for growth, in preparation for division. So when a cell is in G1, it's not thinking about mitosis, the cell is doing its normal stuff. So that's the big difference between G1 and G2, even though they both have a lot of growth in them. G2 is growth in preparation for mitosis. G1 is growth in terms of cell doing its everyday job, gets a signal to divide, and then DNA is going to replicate, and then we're going to go into G2, the growth phase in preparation for mitosis. And the cell's going to grow more so that we can have enough organelles and proteins and membranes for the two daughter cells to have everything that they need. To talk for a second about the part of interphase that we're calling GO or G0. So when cells don't need to divide and they're just going to hang out is when they're in this G0 phase. So this is a cell that isn't thinking about dividing. It's going to continue doing its everyday job. It's not going to go through the rest of the cell cycle. It's just going to hang there. So that's the G0 phase. And we'll talk about how those signals occur to push a cell through the rest of the phase in a second. All right, so, oh, there we go. All right, so what does a cell look like when it's hanging out in an interphase? And we'll look at these under the microscope too. So during interphase, the nucleus is well defined, meaning that we can see the membrane, and the DNA is just loosely packed in long chromatin fibers. So that's what the two pictures are showing us here. Okay, and so this looks like the picture from your book. We have a little dense area inside called the nucleolus. The nuclear envelope is intact with its little pores. This is the chromatin fibers that are here. Okay, and we can be preparing for mitosis. All right, so this is before we're going into prophase. Right, so chromosomes replicated, the DNA and the proteins, and we produce proteins and organelles, all in preparation for mitosis. So you've got to have DNA, you've got to have all the organelles, and whatever proteins you need. Okay, so the S phase, copying and replicating DNA. So it's called the S phase because it's the synthesis phase of interphase, and the dividing cell, the parent cell, is going to replicate its DNA. And it's got to separate the DNA copies correctly into two daughter cells. Okay, and just as a point of reference, the human cell duplicates approximately 3 meters of DNA, and each daughter cell gets a complete identical copy. Right? And the rate of error that can occur with mitosis in DNA replication is about 1 per 100 million bases. So there's a small amount of error. And there are 3 billion base pairs in the mammalian genome. And of course, we've gotten really far in figuring out all of the genome at this point. And there's approximately 30 errors per cell cycle, which can result in mutations to somatic body cells. But we've got checkpoints to take care of this when it occurs for the most part. And when we don't have a successful checkpoint, there's a serious result, and we'll talk about that in a little bit too. So how is DNA organized? What is the difference between just plain DNA and a chromosome? Right, so that's what we're going to talk about here. And what page is this in your book? So I try to remember. It's on page, well, it's not great in your book. 219, 220. I think DNA is elsewhere in the book too. But all right, so DNA is organized into chromosomes. Why? Because if we're going to be taking genetic material and taking it from a parent cell into a couple daughter cells, it would be a lot easier if DNA is organized as opposed to being all loose in the nucleus. And of course, we know that DNA is a double helix molecule. So there's the DNA, and it is wrapped around histone proteins. Okay, so that will be the next level of organization here. Okay, and this is all to compact it to take long, long, long strands of DNA and get it compact and organized so that it can be easily divided up. All right, it's like threads on spools. That's a great way of thinking about it. So the DNA and protein complex is what we call chromatin, okay, which is organized into a long, thin fiber, and that's what we're seeing there. Okay, so we've got the DNA and then wrapped around histone proteins and organized into the long, thin fibers called chromatin. And then it condenses further during mitosis. So you take these big long threads and then organize them, and then this is what you're used to seeing when you think of a chromosome, and this is a duplicated chromosome, okay? A double-stranded chromosome, okay? So this is after it's been duplicated. Okay? After the DNA is duplicated, the chromatin condenses. Okay, coiling and folding to make a smaller package, to keep it compact and organized for separation. Okay, so this is the DNA, the helix that you're used to seeing. Okay, there are the histones and the chromatin, and here's the mitotic chromosome, the duplicated chromosome. So if you're kind of going backwards, this is kind of the chromosome you're used to seeing. Well, what part of that is DNA? Well, let's look at this tiny part of it and blow it up. Well, we've got this chromatin, this tight helix chromatin. If we look at the chromatin closer, we see that it's DNA wrapped around histones, and if we took out those histones to look at the DNA, well, that's where we see the double helix DNA molecule. Does that make sense? So it's a way of organizing the DNA and keeping it protected and intact. Okay, so this is showing you moving from interphase, where we have the chromatin, into prophase, where it's more condensed, and this I'm sure you're used to seeing is the prometaphase, where we're seeing the duplicated chromosomes, and these are the mitotic chromosomes. I always found this really confusing, so you might want to spend a little time, looking at this image and understanding the level of organization. Okay, so here's a SEM image, the scanning electron microscopy of chromosomes. So they really, really do look like those little X's. So these are mitotic chromosomes, duplicated chromosomes. So taking apart that chromosome, okay, we starting out with this kind of half a chromosome. It gets duplicated and held together by the centromere, okay, and then is separated to go to the daughter cells. Okay, so these are called chromatids. They are sister chromatids. So two chromatids make a chromosome. Two chromatids are joined by the centromere. So it's two sister chromatids, narrow at the centromeres, okay, contain identical copies of the original DNA. All right, and those are homologous chromosomes. Okay, homologous means the same information. Homo means same. All right, so here they're single-stranded, and then the homologous chromosomes are here. All right, as double-stranded, those are sister chromatids joined together at the centromere. Okay, so mitosis, dividing cells' DNA between two daughter nuclei. And I think this is a great phase. It's the dance of the chromosomes. It has 4 phases. There are kind of remember that there's 4 distinct phases, but it's one big process. It's one big dance. It's in constant motion. Prophase, metaphase, anaphase, telophase. Paul made a taco. Okay, so let's go through the phases. Prophase. Chromatin condenses. So the chromosomes become visible, and we mean visible under microscopy. You can easily see this with a regular light microscope with a little bit of dye to assist you. Okay. You can see the chromatids. All right, and then the centrioles move to the opposite poles of the cell. Why? Because they're going to aid in supporting the spindle fibers to pull the chromosomes apart. Okay, so the protein fibers across the cell form mitotic spindles. Those are like, I mean, you kind of call them strings, but they're much more firm than that. They're big fibers. Okay. And then the microtubules, the actinomycin that we talked about earlier, and we're coordinating the movement of the chromosomes. And the nucleolus disappears and the nuclear membrane starts to break down. Why does the nuclear membrane start to break down? Well, we're going to want to take, going from a cell where we've got all the DNA in this nucleus, and then we're going to somehow make 2 cells, so we're going to need to end up with 2 cells with their own nuclei. Well, we've got to get the DNA out of the nucleus, organized and separated. So that's what's happening here. We are condensing the chromatin, okay, the centrioles are moving apart. We're forming the spindles and breaking down the membrane, all so that we can get the chromosomes out of there, so we can organize them and divide them. Okay, and then it gradually moves into metaphase. So the space between prophase and metaphase is called prometaphase. So during this time, we have the spindle fibers attaching to the centromeres. Okay, so that's what this picture is showing you, this transition. Okay, creating kinetochores. And kinetochores are a protein complex. So I switched PowerPoints here to just go over kinetochores in a little bit more detail. They're part of the structure here, and this is, kinetochores have a role in prometaphase. So some of the microtubules attach to the kinetochores of chromosomes and begin to move the chromosomes. So they're protein complexes attached to the centromeres. And at metaphase, when the chromosomes are all lined up at the plate, the metaphase plate is an imaginary structure. So let's just, across the middle, let's just show you a picture of the kinetochores here. Excuse me. All right, so here we're looking at a little bit closer to metaphase, but the kinetochores are right here near the centromere, so it's a little complex. And then these microtubules are called kinetochore microtubules attaching to the kinetochores. And the reason they're emphasizing that the metaphase plate is imaginary is because there's no structure across the middle. It's just, again, we're, lining up across the middle as a way to start organizing the chromosomes in preparation for division. So there are some microtubules that are associated with the kinetochores and some microtubules that are not. So those are the non-kinetochore microtubules. And why do we care about kinetochores? Well, it's just an interesting part of how these things are moving. So it's a part of that structure. So these are the sister chromatids, okay, and the centrosome sitting at the pole of the cell, and this complex is called the astar. And then here's just a little close-up of that scanning electron microscopy shot of the kinetochores and attached to the microtubules. Kind of cool, right? This is why we keep saying again and again about proteins being important in the structure. All right, so I'm gonna switch back to the other PowerPoint. I'm not going to pause because it's easier if I don't. And there we go. So fancy. All right, so transitioning to metaphase again. So just to show you, so here's the kinetochore and the non-kinetochore microtubules, and of course the microtubules attached to the kinetochore are called kinetochore microtubules. And I hope I'm saying kinetochore right, but I'm going to say that I've never heard that word said out loud. It's just something I've read a bunch of times. So hopefully Miss Aber will correct me if I'm butchering that word. All right, so prometaphase, the spindle fibers attached to the centromeres, creating kinetochores. Right, microtubules attach at the kinetochores, what we talked about, and connect centromeres to centrioles. I think it would be really excellent if you're getting some of these terms confused is to make yourself a few flashcards. And when I do the review video for mitosis, I'm going to be emphasizing again things like knowing the difference between a centromere and a centriole. All right, so then the chromosomes start to move. Cool, right? I think it's cool. All right, so here's a really cool dyed image showing you prometaphase. My dog is laying next to me and snoring, and I hope you can't hear her, but you probably can. She's snoring like crazy. Okay, next. Metaphase, chromosomes lining up along the middle of the cell. Why? Because they're organizing themselves in preparation for division. Right, so they're along this imaginary metaphase plate. There's no real plate sitting there. It's just the middle of the cell, like the equator of the cell. Meta means middle. Right, so the spindle fibers coordinate the movement. Right, so we're all lined up and organized to start pulling the sister chromatids apart. We want to make sure that separation happens perfectly because that's critical to ending up with nice normal daughter cells. Right, so we are ensuring that the chromosomes are going to separate properly so that each new nucleus gets only one copy of each chromosome. All right, so the centriole pair is up there, this centrosome and the aster. So this is giving you kind of a summary of what we're talking about. It's showing you the imaginary metaphase plate, okay, and the kinetochore microtubules are going to be jerking the chromosomes. The kinetochore of the sister chromatids are attached to the kinetochore microtubules coming from the opposite poles of the cell, and where the spindle, of course, is the entire apparatus of the microtubules because of its spindle shape. That's why it's called that. Metaphase is the longest phase of mitosis, probably because this lining up is a little bit involved in preparation for pulling apart, because this is a critical point where you don't want any errors. All right, so when we look at it under the microscope, you're going to see really clearly how you can see the spindle and complex and the chromosomes lining up there. So, you know, it's not like they're sitting in the equator all perfectly flat, but you can see how they're lined up there. And this is showing you the kinetochores with the microtubules attached to them. Okay. Oops, there we go. Anaphase. Okay, sister chromatids are separating at the kinetochores, moving towards the opposite poles, pulled at the centromeres. Okay, so these are now daughter chromosomes being pulled apart. Okay, they're pulled by motor proteins walking along the microtubules. So if you can imagine that inner life of the cell video, we saw that they're literally can be walking along. I think I have a little animation of it I'll show you at the end. Okay, the actin and myosin proteins are having a role here. We'll talk about that in 1 second. And we have an increased production of ATP by the mitochondria. So the actin and myosin are having almost like a muscular role in that movement. And this is referring again to the picture on 112 from chapter 6 that goes over how the parts of the cytoskeleton work and the actin being a part of the motion inside the cell. And the poles are moving farther apart, so that we're getting, moving towards being two separate cells. So these are getting a little bit further apart, and the chromosomes are formed into what am I trying to say? The chromosomes are becoming the form that they're going to take when they're in the full daughter cells. It's hard to come out in full sentences all the time in these videos, I gotta say. Okay, so the polar microtubules are lengthening. So that's a part of how, when the poles move further apart, the microtubules are going to lengthen. Phew! Okay. All right, separation of the chromatids. Okay, so this is what we're talking about when we're talking about anaphase, is about the separation of the chromatids after they've lined up along the metaphase plate in metaphase and they start to separate. Okay, this is what's happening. So the proteins that hold the sister chromatids together are inactivated. All right, they separate to become individual chromosomes. So this picture is one chromosome, two chromatids. All right, it's a double-stranded, and then it transitions, okay, to being two chromosomes that are single-stranded. Okay, so this shape and this shape are still chromosomes, okay, but this chromosome is composed of two chromatids, and this chromosome is one chromosome, and this chromosome is another chromosome that are single-stranded. So when we say chromosome, we don't necessarily always mean this, right? And that's a critical thing to remember, that students often get confused by, that a chromosome always means this, that it's the two chromatids together. But no, here we've got two separated chromosomes, right? And here again, we're showing the kinetochore and kinetochore microtubule complex that's involved in this motion, the aster, the polar microtubules, and the centrosome. Okay, kinetochores use motor proteins that walk chromosomes along attached microtubules. And by walking, we mean that the motor proteins actually look like little feet, and they move along using ATP along the way. So the microtubule shortens by dismantling at kinetochore, the chromosome end. So this is from your book on page 225. That's the experiment that's being discussed there. So the experiment is about, how did they figure out which end was shortening, and they used fluorescence to see, and the conclusion was that during anaphase, the kinetochore microtubules shorten at the kinetochore end, not the spindle pole ends. and that's how they figured that out. So I would definitely read over that experiment so that you can understand this movement during anaphase. So the microtubules are not reeled into the centrioles, which you may have learned because that's an old way of thinking about it. It's not like a fishing rod. Instead, the motor proteins walk along the microtubules like little robots on a clothesline, like walking along. In dividing animal cells, non-kinetochore microtubules are responsible for elongating the whole cell during anaphase, getting ready for cytokinesis. Okay, moving on to telophase. Okay, chromosomes arrive at the opposite poles. Okay, so the chromosomes have fully moved over to one side and the other. The daughter nuclei form, meaning that we start to get the nuclear envelope to form. We start to get a nucleolus and this cleavage furrow. So nuclei form, the chromosomes disperse, so they're no longer visible under a light microscope. All right, the spindle fibers disperse, and we have the beginning of cytokinesis and full cell division. Okay, so we'll talk about cytokinesis in a second. Okay, cytokinesis in animals, we get this is the cleavage furrow we're going to talk about, the constriction belt of actin microfilaments around the equator of the cell. So how are we dividing the cytoplasm and ending up with two distinct cells with the membrane around them? Is that we're going to have the actin microfilaments forming a belt in there. And remember, the actin is kind of about movement, and is a part of how muscles work. And we'll talk about actin and myosin and muscles. So actin are creating a little constriction in here. And we start to get a cleavage furrow, okay? So the cleavage furrow is a groove where the metaphase plate was, okay? So you have actin and myosin contracting here, okay? And the cell splits in two. It's like tightening a string around a balloon, okay? Or some other big ball of goo or something, okay? And all right, so there you get a little animation of mitosis. All right, and there's a close-up, the scanning electron microscope picture of the cleavage furrow. So you can see it's like a belt pinching in. Okay, and we have a contracting ring of the actin and myosin complexes here to result in daughter cells. If you've never been to the website Cells Alive, it's cellsalive.com. It's a really great place to kind of review the basics, and they have a lot of really cool animations like this to help you visualize mitosis and meiosis and other things. Okay, so this is one of the types of organisms we commonly use to look at mitosis in an animal cell. It's a whitefish, and we have these slides in the lab, so we'll be using these too to help you visualize it besides just looking at a picture. So this is interphase, prophase. What's happening there? What do you see? All right, what's happening in metaphase? All right, this has been dyed, of course, to show us the chromosomes nice and dark. Okay, and here's the beginning of anaphase, and then early telophase and late telophase. All right, so you can see that we've got the distinct membrane happening there. The chromosomes are organized into new nuclei. And here's cells that have been dyed with fluorescence. Really cool looking. So here's interphase. We can't see the DNA or the chromosomes at all. Here we have the condensing chromosomes. The nuclear membrane starts breaking down. All right, between prophase and metaphase, here we see the super condensed chromosomes. Here they are lining up across the metaphase plate, and you can clearly see the microtubules here. Anaphase, the chromosomes pull apart, and telophase, the new nuclei begin to form, and you can see that pinching of the cleavage furrow. All right, cytokinesis in plants is a little bit different. All right, we need to have a cell plate that forms, because remember that we want to have two cells that both have cell walls. And so the vesicles line up along the equator here. Right, they're derived from the Golgi apparatus. The vesicles fuse to form two cell membranes. It's a cool use of a membrane-bound organelle. And the new cell wall is laid down there. And the new cell wall fuses with the existing cell wall. And so these are the vesicles lining up there, forming a cell plate, two daughter cells, and we end up with a cell wall. And here's a microscopic picture of it close up. So you can clearly see the vesicles all lined up there, right? And vesicles, remember, are surrounded with a membrane, so that's how they're going to form the membrane. So here's mitosis in a plant cell. And we commonly use onion cells to look at mitosis. So here's that. I would like you to definitely pause the video and see if you can locate all the phases of mitosis and count how many there are in each phase, because that is one of the things that we'll be doing in the lab. So I would like you to pause at this point. I'm not sure if it'll pause clearly, but if you can, pause it and count how many cells there are in each phase: interphase, prophase, metaphase, anaphase, and telophase. And let's see, yep, you can definitely see all the phases here, and practicing that skill will make the lab much easier. Okay, so the evolution of mitosis in eukaryotes evolved from the binary fission of bacteria. So if you look in your book at the section on the evolution of mitosis, which is somewhere, where is it, page, oh, I can't find it quickly. Oh, there it is. Page 227 is the evolution of mitosis, and in figure 12.11 shows you bacterial cell division, and you are responsible for understanding bacterial cell division. So, which is obviously a little bit different since bacteria don't have a nucleus. So we have the origin site of replication. Chromosomes are replicated, and one copy of the origin moves rapidly towards the other end of the cell, and the cell elongates. Okay, and you have this ring of proteins forming down the middle, and the cell pinches in two. Of course, there's just a single circular chromosome and no membrane-bound organelles, so we're going to end up with the circular chromosome in the replicated cells, just like the origin. And this, of course, in prokaryotes can happen at an extremely rapid rate. Okay, so this is very similar to the picture in your book, showing you prokaryotes, protists, a couple different kinds of protists, eukaryotes like yeast, and animal cells. So in bacteria, there's no nucleus, there's just that single circular chromosome, and after DNA is replicated, it's partitioned in the cell. After cell elongation, the FTSZ protein assembles into a ring and facilitates separation and cell division. And you can look at that in your book too. Protists have a nucleus, and a nuclear envelope remains intact during cell division. The chromosomes are more linear, and So there's many different kinds of protists, so they work a little bit different between the dinoflagellates and the diatoms. All right, and then in yeast, the nuclear envelope also remains intact. A single kinetochore microtubule attaches to each chromosome and pulls each to a pole. And then, of course, we know how the animals work. So you can see the relationship between prokaryotes and some of the single-celled eukaryotes and the animal cells. So here are the algae, okay, showing you, I don't think these are in your book. No, these are not in your book, but just to give you a picture of them in the process of division. Here's the diatoms, which are really cool microscopic algae found in water. They're really just beautiful. Okay. Any questions? You can definitely stay after with Miss Aber and ask her some questions if I'm still out. This one was long. The next one's going to be on control of the cell cycle.